have a poem that regards the mysterious process and powerful tool that are transformed cells. Most human cells can't live outside the body or have a very limited shelf life. But these transformed cells can live in a petri dish or flask and divide apparently indefinitely, forever in the laboratory. I love cell lines and could not resist writing a poem about them. In fact, I have a few more in the hopper. They provide an amazing gift to science. We can treat these cells in a petri dish with chemicals and conditions that we just can't do to a human. We can learn so much from observing and testing these cells. How they acquire this amazing ability to divide continuously is still mysterious, and I can't wait to talk about it. But first, the poem. Transformed. What was heard from the viral whispers that made you haunt Hayflick? Capsules for the continuous commune. Reborn without belly buttons, the undead pushed into your script. No arrest, paroled to the incubator crypt. It's hard to look at the human form and understand all that's happening within it. Lenses were crafted to peer into life's secrets. If it could just be seen, it could be known. That was what we thought. We now know it's much more complicated. But for every major scientific tool, the surmising that this new thing was going to solve everything is commonplace. We're not sure who the first persons to study cells were. I mean, let's be serious. Imagine all those curious kids playing with lenses in the 1600s. They may have been the first one to discover a cell, but they didn't get a Nobel Prize for it. Anyway, in the mid-1600s, looking at slices of the world under multiple lenses to really focus in on details became a thing. These early cell biologists, as we now call them, used stains and dyes to try and better see boundaries and see what was going on down there. What are these little bubbles doing? But until 1960s, most of these tissues on the microscope stage eventually died or were already dead or were killed by the toxic interventions that people were using. And even without anything toxic, every care and coaxing trick employed, people noticed that cells just couldn't live after about 50 cell divisions. This limit of the number of cell divisions is called the Hayflick limit. Why Hayflick, you ask? Because one of the people involved in the noticing of the cell death wall was called Hayflick. So, in 1962, Henrietta Lacks was treated for cervical cancer. Without her consent, her cervical cells were placed in an environment that allowed them to continue living if they were able. This had been done many times before, and every time the cells eventually all died out. Not this time. Given fresh fluid to bathe in, Henrietta's cells lived, dividing, metabolizing, signaling, replicating, growing. We shall come back to the ethics of not only the origin of cell lines in general, but many more issues in other episodes. Sometimes we learn things from really bad mistakes and errors in ethics. And it's confusing because on the one hand, there's deep gratitude to the knowledge gained while pledging never to use that methodology again. Mrs. Lack's cells did something unseen and unknown prior to 1962. They grew quite happily outside of their host, growing and dividing indefinitely. In fact, this cell line, HeLa, the descendants of those first cells, are now in laboratories all over the world right now. When cells acquire the ability to live outside their host, they're labeled transformed. What causes this breaking away from what is referred to as Hayflick limit? Well, a genetic alteration seems to be the source of all of the changes in cell lines cataloged thus far. Mutations to turn off the cues for senescence and cell death, and the turning on, or the ability to buy more time with genetic additions. Telomeres are like caps at the end of a DNA molecule packaged tightly as a chromosome. You can think of them as long strings of tickets that you can get at a carnival. At the beginning of the night, you start out with a lot. And then as the night wears on, you can see that your night of fun will come to an end once the tickets run out. This is similar to how the shrinking telomeres at the end of the chromosomes signal the march closer to the final division. But what if you could game the system and get a whole bunch more tickets? You'd be set for a while longer. After a cell has transformed, it can activate telomerase, an enzyme to add more telomere genetic material to the end of the chromosomes. You know that one skee-ball machine at Chuck E. Cheese that's on the fritz and just keeps printing more tickets? Yep, that's telomerase, providing more tickets for more license for more cell divisions beyond the Hayflick limit. It has been observed multiple times that inoculation or transduction of a cell with specific viruses can make this magical switch to transformed cells happen. The cellular fountain of youth. Capsules for the continuous commune, meaning entry and overtaking of the normal cellular processes that safeguard the cell from living beyond the usual number of cell divisions. Viruses, their genetic material encapsulated in protein, taken in by the cells in tissue culture. This fusion of capsules, the virus and the cell, brings new genetic material into the cell that hadn't been there before. And sometimes when this happens, the genetic material can insert into the cell's DNA in the nucleus in a way that changes the cell's genetic instructions and therein behavior. Many cell lines that exceed Hayflick's limit are transformed or changed similar to this. A new piece of genetic instruction from a virus changes the way that it behaves and allows it to divide forever. Dividing past this limit is less than ideal for an organism because mutations accumulate. But these cells, once they become transformed, they enter a new way of being, a rebirth. And then, of course, I joke about the belly buttons, as they are only cells. Yet in that nod to nutrition and waste exchanged, separated from their host, these cells need a fresh and frequent replenishment of what an umbilical cord would do. It's left to laboratory staff and students to ensure that the cells have enough space, food, and waste removed. They have to be at the perfect temperature, humidity, and CO2 percentage. Cell cycle arrest is a timeout. cells go into a sort of hibernation when food is low or oxygen is low. I've accidentally forced my cells into cell cycle arrest when I misjudged how much media or food to bathe them in until the next time I checked on them. And when I came back and the media had turned from red pink to orange yellow, it has a pH indicator in it. And the yellow indicates more acidic because the waste products were accumulating and it smelled a different kind of funky. Under the scope, the cells look sad. Those that were merely in timeout were barely hanging on and had shut down all but the most essential functions, hoping I'd get back in exchange for their food source. I say this to provide more context for the word arrest. In the world of cell biology, it means something different than, say, in law enforcement. And other than my recent confession of poor custodian of cell lines, my cells did not often experience nutritional deprivation that caused a pause. But sometimes we intentionally deprive nutrition so that all of the cells in the petri dish pile up into the same state of the cell cycle, so they're synced. Another poem for another time. So at last, the last line of the poem, no arrest, refers to the possibility of continuous cell activity and division and the cycling without pause. However, the next segment describes the destiny of the transformed as anything but blissful freedom, rather a life without parole, kept warm, quiet, and forever in the incubator. Now let's read the poem again. Transformed. What was heard from the viral whispers that made you haunt Hayflick? Capsules for the continuous commune. Reborn without belly buttons, the undead pushed into your script. No arrest, paroled to the incubator crypt.